All right, we can go ahead and get started. Thank you everybody for coming to the February StaPH-B call. Today we have Spencer Bruce with us from New York, and he'll be talking about some more practical approaches to using bioinformatics software in public health. Spencer, if you're ready, you can go ahead.
Great, thank you. So I don't know, my video is kind of weird. I don't know if it doesn't seem like anybody can see me. It's just black, right?
Mm-hmm.
So hopefully I can share my screen. So I'll do that now. But I apologize for that. I don't know. It's something strange with Zoom in particular.
No
Um,
worries.
so let's see. One second. Okay. Is everybody able to see that?
Yeah, that looks good.
Great. All right. So, hello, everybody. I'm Spencer, and thanks for having me today. I'm the director of the Bioinformatics Core here at Wadsworth, and I've been here for a couple of years now. And in my position, I really work to support our bioinformaticians and help drive the strategic decisions about how we do things here. Um, I'm not in the weeds writing pipelines so much myself anymore, um, but I think I was hired to also kind of fill this back end computer science gap a bit. As in my previous position, I managed the HPC resources at the University at Albany. So my background is really both bioinformatics and high performance computing. And as all of you know, as bioinformaticians, we rely very heavily on open source software. So today's talk focuses on practical ways to use, manage, and reuse that software under the real world constraints that we all have working in public health. So the goal is not to promote a single tool, but instead to provide a framework for um, making informed choices. So open source bioinformatics software is powerful, but it's also kind of fragile. Small changes in versions or dependencies can affect results. In public health, analysis often need to be reused, audited, or shared, which really makes reproducibility critical. I think Wadsworth is a little different than many other public health labs because we have both a clinical side where we are producing pipelines to inform clinical decisions related to common infections, et cetera. And we have a research side here where we do a lot of one-off work to support PIs who have various research endeavors. And that one-off work usually means creating some sort of bioinformatic code for a peer-reviewed publication or for a pilot project or for a grant. And I think many of us working in bioinformatics in public health come from the academic world. And that academic world is more PI based. So when we were in graduate school or undergrad, or when we were postdocs, we probably did a lot of bioinformatic work that was also one off. We weren't expecting to have to reproduce it year after year and make changes and keep using it. So there seems to be a learning curve with a lot of people that come into bioinformatics and public health as it relates to that reproducibility aspect. And that reuse and reproducibility really matters for the type of work that we do. So our work in public health involves managing change. We create a pipeline to address a certain pathogen, And then over time, we have to make slight modifications to that pipeline to account for new tools, new sequence types, new AMR genes, etc. So not only are we interested in reproducibility, but we need to make changes to these reproducible things as well. Additionally, we often have multiple valid technical approaches to address a problem, which makes this more confusing because we're asking ourselves the question, do we use this open source software package or do we use that open source software package? And on top of all of this, one of the things we have to deal with in public health is operating under different IT security controls, infrastructure availability, staff expertise, and time. So there are a lot of things controlling how we do things that are kind of outside of our control. And I think that one thing that makes a centralized approach to public health bioinformatics so difficult is that every public health lab is working under these different constraints. So the most basic constraint that affects how we work is the OS we are working on with our machines, right? Bioinformatics commonly runs on Linux. It's essentially its native platform, if you will. We can still theoretically do bioinformatics on Windows using something like the Windows subsystem for Linux or Mac OS, because Mac OS is very Unix-like, although there are some compatibility issues. So for many of us in public health, we are likely using Linux virtual machines that are set up by our IT department or configured in the cloud. So given all that, before we even move forward, I'm interested, maybe we can raise some hands and see how many people are doing their bioinformatics. Well, let's start with Windows, the least likely. How many people are doing bioinformatics on Windows? I don't know. I don't see any hands here, but maybe it's just Zoom and I don't have it set up correctly. Well, I can't see it, so I don't know. I don't know if anybody sees something I don't and wants to shout it out. Well, I guess not. So if everybody wants to sit quietly and listen, I guess we can go that route too. Um, oh, here, I have some things in the chat. Okay, so we have people saying, um, I use Windows subsystem for Linux and Windows, okay? Okay, great. Okay, so I guess people do use Windows, right? Windows subsystem for Linux. Genius on Windows is nice for a quick viz, so some people are using Genius, etc. All right, well, that's good to know. So I guess if we do this again, we'll have people respond in the chat. Well, then let's have people respond in the chat. Who's using Mac OS to do their bioinformatics, public health bioinformatics? Anybody? No one in the chat. So I'm assuming that there's probably many people using Linux. How about Linux? We see some people using CLC Genomics Workbench. Linux for most things. Okay. Some web-based pipelines, local Linux, Ubuntu in cloud, Linux on HPC, Linux workstation. Okay, cool. So we have people using all different things, right? It's not, and that makes our lives hard, but that doesn't mean we can't share things or we can't work with each other. So, as we move through this, I'm going to talk a little bit about the things we can do to get better at sharing, even though we're kind of working in different environments in different ways. So, most of this presentation is going to be Linux based. I think that's the OS that everybody here uses for our bioinformatics. And if you are Linux based, uh, there are some basic software installation methods that you can use in a Linux system, right? You have apt-get brew. Both of these, apt-get on Linux and brew on Mac OS, are system level package managers. When you install software this way, it's installed for the entire system, and it's shared by all users. The big advantage here is simplicity. These installs are usually easy, stable, and well tested. If you need a basic utility or a common tool, this is often the fastest way to get it. The downside is that versions are limited to what the operating system provides, and any change affects everyone, right? So if you update a tool to support one analysis, you might accidentally break another analysis that depends on the older version. In shared environments, especially in state labs or HPC systems, this is why these installs are often tightly controlled by IT on these shared resources. We also have things like pip. So pip is the standard package installer for Python. It's fast, flexible, extremely popular because so many bioinformatics tools are written in Python. And the strength of pip is that it gives users a lot of freedom. You can quickly install packages, experiment with new tools, and iterate rapidly. The downside of pip is that installs can easily lead to these dependency conflicts, especially when multiple tools require different versions of the same library. And without careful isolation or version pinning, pip environments can drift over time, making it hard to reproduce results or rerun analyses months later. The other option you have from a basic standpoint, right, is to install from source. So installing software from source means downloading the code and building it manually. So this gives you the most control over versions, compiler options, and system integration. And in some cases, especially for specialized bioinformatic tools, this may be the only option. However, this approach is time consuming and it requires lots of expertise, right? More importantly, it's a difficult process to document and reproduce down the line when others might need to use these tools. So if someone else needs to rerun your analysis, or if you need to rebuild the software on a new system, it can be very hard to recreate the exact same setup. So the big picture takeaway here, right, is all three of these methods work, and they're often the starting point for bioinformatics work. But they tend to break down as soon as you have multiple projects, multiple users, or a need for reproducibility. So the rest of this talk is really about what we do after these methods stop working for us, stop scaling for us as we move forward with our bioinformatic work. So now I'm going to talk about Conda. So I'm interested again, if people want to reply in the chat, are people here using Conda to manage their software? Yes, no, maybe so. Yes, I have a yes. We use it for some stuff. That's great. Okay, cool, right? So, okay, Conda. So Conda is used. That's great, right? So Conda emerged because those basic methods we just discussed don't scale well once you have multiple projects, tools, and people sharing the same systems, right? So what does Conda give us? It gives us these isolated environments, and that's really what Conda is all about. Instead of installing software directly onto the system, Conda lets you create self-contained environments that include specific versions of tools and their dependency. This means software used for one project doesn't interfere with software used for another. With Conda, it became normal to think in terms of one environment per project. And it sounds like in the chat here, that's how a lot of people are using it. And I think that's great, right? So each project can have its own environment file that describes exactly what software it needs. This makes it much easier to switch between projects, pause work, and come back later without worrying about what changed on the system. This separation is really about preventing interference. So without isolated environments, installing or updating a tool for one analysis can silently break another. Conda environments reduce that risk by keeping tools scoped to the project they belong to. So many bioinformatic tools are packaged and maintained by the community. Conda is as well through things like CondaForge and BioConda, which a lot of you might be familiar with. This made it much easier for researchers and public health labs to access complex tools without building everything from source. So the diagram that you're looking at on the right here helps to clarify a little common confusion. So Conda is really the underlying package and environment manager, where mini Conda is really a minimal distribution that includes Conda and Python. And then we have Anaconda, which is this larger distribution that bundles Conda with many pre-installed packages. So for many public health labs, the lighter weight options like Mini Conda or Mini Forge provide more control and fewer complications. So, the reality is Conda didn't just give us a new tool. It kind of changed how we think about managing our software. Instead of treating the system as the environment, we started treating each project as its own environment. And that shift really laid the groundwork for everything we'll talk about next, including containers and workflows. So the other thing I want to talk about a little bit is also kind of the downsides of using Conda. So there are some major complications with Conda that emerged more recently, right? And it's not really a technical limitation, but it's a licensing one. And what I've found in talking to people is a lot of people don't even know about this, right? So in the early 2020s, in the last 3 or 4 years or so, Anaconda Inc., the company that maintains the Anaconda distribution, updated its terms of service. So under these updated terms, certain types of organizations, including large organizations, commercial entities, and government agencies, require a paid license when using Anaconda's hosted package repositories at scale. So this is obviously especially relevant for public sector organizations like state health labs. Even if individual users are not doing anything unusual, centralized or large-scale use of their servers can fall under this licensing requirement. My understanding too is that they do occasionally ping where people are pulling packages and send letters to them telling them they need to buy a license if they want to keep using Anaconda in that capacity. So this introduced a lot of uncertainty for government labs that rely heavily on it. One of the most important points to emphasize is that Conda itself remains open source, and you can still use Conda software freely. What really changed is licensing around downloading packages from Anaconda's own hosted servers, especially at scale. And that's, if you go back to the last slide I was showing you, that's the Anaconda larger universe. So this has caused tons of confusion, really. It was very confusing for us here when we were trying to figure out how to deal with it. So this issue is really about which repositories your software is downloading packages from, not the Conda tool itself. For state or government IT, even this sort of perceived ambiguity can be a problem, right? Because the IT department starts going, whoa, 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 because it raises questions about compliance, procurement, long-term support, etc. So, this is a problem that a lot of people are having to deal with now. But the good news is there's kind of an alternative that essentially lets you skirt these Conda licensing rules, right? So the most important point again, right, is that Conda itself remains open source. That has not changed, and the licensing changes we talked about apply specifically to Anaconda hosted package repositories, not the Conda software itself. So if you're familiar with using Conda and you're just downloading a package and you're specifying it's from Bioconda, that's usually fine. But if you're not specifying where you're pulling it from, and if it's coming from Anaconda, then you're going against the rules. So MiniForge, which is in the slide here, right, is a lightweight installer that gives you Conda without connecting to Anaconda's servers. And it works just like Miniconda. The installation is essentially the same, but it ensures you're not violating any licensing rules, right? So instead of pointing to the Anaconda-based repositories by default, MiniForge is configured to use CondaForge, which is the community-run package ecosystem. And you can still also access Bioconda using MiniForge as well. So for public institutions like state health labs, the biggest concern is often this licensing ambiguity. Using MiniForge helps to avoid that ambiguity by relying on community-run repositories rather than the commercial hosting services. And you will see now too, if you go to major universities are all well aware of this. And you can see on their how-to pages, they have instructions on how to install and use MiniForge, and they've replaced the Conda pages with them because of these licensing rules. Right, so I think Curtis here says Conda installs channels default pulls packages from Anaconda, which I believe is correct. And I think MiniForge removes channel defaults upon installation. You can also configure Miniconda or regular Conda to remove channel defaults, I believe, as well, although I haven't. It just seems easier to switch to MiniForge, right? And not have to worry about if you're doing it or not, but I believe Curtis is correct. So there are some other downsides to Conda as well. So far I've talked about Conda as a very powerful and widely used tool, and that's still true. This slide focuses on some of its technical limitations, and that's not really as a criticism, but to explain why people eventually looked for additional layers of isolation. And one important thing to understand about Conda is that it still relies on the underlying operating system where you have it running. So even though your Python packages and tools live in a Conda environment, they're still interacting with system libraries provided by the host OS. That means the environment is not completely self-contained. So this leads to a very common situation in bioinformatics, and everybody probably knows this. This is what people describe as the works for me environment, right? Two people can have the same Conda environment file, but if they're running on different operating systems or even slightly different versions of the same OS, behavior can change. And these differences are often subtle and hard to diagnose, which makes them especially frustrating, right? On top of that, operating system upgrades can break existing Conda environments. So in an environment that worked perfectly last year may stop working after you update the operating system on the computer you're working on or on the virtual machine you're working on, etc., even if you didn't change anything inside Conda itself. So this is particularly relevant in managed environments where OS upgrades are outside the control of individual users. I certainly can't speak to everybody in this meeting, but I know here at Wadsworth, we go home at night, and when we come in the morning, our computers have been updated, unbeknownst to us, right? So this creates challenges for long-term support, validation, and auditability. So in public health and regulated settings, reproducibility over time is often just as important as getting results quickly. If you need to rerun an analysis years later, relying solely on Conda is probably a bad idea. These limitations don't mean Conda is a bad choice. It just means it doesn't solve every problem by itself. The one thing I haven't mentioned, and I will mention, is the one big thing that Conda has going for it, whether you're using something like Miniconda or CondaForge, is that you don't need any administrative privileges to install it and run it. You can do it all yourself, from installing it to creating environments and installing open source software. So if you're in a public health lab where you have no administrative privileges on anything, Mini Conda or Mini Forge will open a world of possibility for you in terms of what you can do. So given these limitations we talked about with Conda, um, this created a situation where users started to ask, how can we solve this problem, right? And this is a big shift in how bioinformatics software is shared and reused. So Docker didn't originate in bioinformatics, but it ended up fitting bioinformatics problems extremely well. So who in this meeting is using Docker to help utilize their open source software needs? Oh, I see a hand up from Jake, Brian, Tony. Oh, I see hands now. That's good. Definitely use Docker, Curtis. Great. So lots of people are using Docker, right? So Docker originally emerged to solve what many of us recognize immediately, right? That the works on my machine problem. So in bioinformatics, where software stacks are complex and dependencies are deep, this problem shows up constantly. And the key idea behind Docker is that instead of installing software into an existing system, you package the software together with everything it depends on, including the operating system. And we're talking about libraries, runtimes, etc. Everything is in this container and essentially quarantined. So the result is a self-contained unit that carries its own context with it. So technically, containers still share the host kernel from a deep IT perspective, but from a user perspective, they behave like fully isolated environments. And because everything the software needs is bundled together, the same container can run on a laptop, a server, or an HPC system, or in the cloud and behave the same way, right? So this dramatically improves reproducibility, and it makes it much easier to share tools and pipelines between people and organizations. So bioinformatics pipelines often involve many tools chained together, sometimes written in different languages and maintained by different communities. Containers are just a natural fit for this kind of complexity because each tool can be packaged with exactly what it needs, right? Without worrying about what's installed on the underlying system. And it didn't just make things easier, it made certain kinds of reuse and collaboration possible that just wasn't possible before, right? I think for a lot of us, we would see our open source software essentially age out as the operating system became newer and newer, but this kind of solves that problem. But at the same time, Docker kind of introduced new considerations around security and system access, system access, which is why its adoption looks different across different environments, depending on these constraints that we all have based on where we're working and our IT support. So let's kind of just look at these two and compare them side by side right now. So at this point, it's tempting to ask whether one is better, right? Docker or Conda. And the answer is that neither is universally better, nor are they mutually exclusive, but we'll talk a little bit more about that later. So they solve related problems at different levels, right? And the right choice depends on what you're trying to do and what constraints you're working under. Conda is fundamentally about flexibility. It lets individual users create and manage their own environments without needing special system permissions. No admin privileges needed. And that might be your only option, depending on your IT department, right? That also makes it very fast to experiment, especially during development or exploratory analysis. Docker takes a different approach. Instead of managing environments on top of an existing system, Docker packages the software together with everything it needs to run. This creates a single self-contained bundle that behaves the same way wherever it's executed. And we all know this because StaPH-B has curated a wonderful collection of Docker containers for us to pull when we're designing pipelines. So an important distinction is that Conda environments still share the host operating system. That's why it's lightweight and easy to use. Docker isolates much more of the runtime environment. So because the container defines the software stack more completely, it is less sensitive to differences in the host system. This is a big reason containers improve reproducibility. So the trade-off kind of looks something like this, right? Conda is often faster and easier when you're developing or experimenting. Docker is often better when you need to share analyses, reuse pipelines, or guarantee consistent behavior across systems. In real-world bioinformatics workflows, many groups use both, right? Conda during development, containers for production and sharing. In state lab environments, this choice is shaped again by your security policies and the infrastructure that's available to you. Sometimes Docker isn't allowed. Sometimes Conda is preferred, and sometimes both are used together in different stages of the same workflow. Of course, though, Docker's stronger isolation does come with trade-offs too. There are downsides to Docker, especially around security and system access, and that's what I'm going to touch on next. Okay, so far up to this point, I've talked about what Docker enables and why it became so influential in bioinformatics. This slide focuses on some of the practical downsides, the things users tend to notice first when they start working with Docker. One immediate challenge with Docker is that it typically requires special permissions, right? You're not going to get away with using Docker if you don't have any sudo or administrative privileges. Unlike Conda, which users can often manage themselves, Docker usually needs additional setup or approval from IT. Because of how Docker operates, it's not allowed on many high-performance computing systems at all. This is extremely common in academic and government environments, including state labs. So when Docker isn't available, it's usually a policy decision rather than a technical oversight, right? Docker also comes with a steeper learning curve. Users need to understand new concepts like images, containers, and build files, which can feel like a big jump for people coming from more traditional bioinformatics backgrounds. So Docker is generally less convenient for rapid experimentation. Making changes often means rebuilding an image rather than just installing a new package, and that can really slow down development. Finally, Docker images can become quite large, especially for complex pipelines. So large images take longer to build, store, and move around, which can be a practical limitation. These are trade-offs that show up from a user perspective, but they're not really the whole story here, right? So, the other side of this story is how it appears to our IT department and from an IT security perspective. Docker typically runs with elevated system privileges. From a security standpoint, this means container processes may have broader access to the traditional user-level applications. Because of this privilege model, Docker can bypass security controls that organizations rely on, and generally you might find that your IT department is scared to death of you using Docker. So this doesn't mean Docker is inherently unsafe. It means it doesn't fit neatly into older security models. And if you work for a state government like mine, you probably have an older IT security model, right? Another concern is that Docker commonly pulls images from external registries, right? Like StaPH-B. So that means arbitrary code can be introduced into an environment unless there are strong controls around image source and validation. And many ID departments now have tools to scan or look to places like Quay.io to see the security vulnerabilities within these containers. So at scale, monitoring container activity can be pretty challenging for IT departments. So traditional monitoring and auditing tools may not integrate cleanly with containerized workloads. Since Docker can conflict with operating system policies designed to restrict behavior, this can make Docker essentially a no-go for many IT departments out there. So these concerns are not a criticism of Docker, really. They're reasons why many organizations say no to it or place limits on it. The other thing I want to mention too is just the last bullet point here, which is may break OS policy rules. So we've been in a situation here at Wadsworth where we've been told IT policy says all operating systems must be fully supported, updated, and patched on a regular basis. And that does not just apply to your computer, that applies to the containers you use as well. So we're in a position where we can use containers, but they require some very extreme maintenance to keep them up to date for that purpose. So given all of these fears about Docker, I'm now going to talk about something called Aptainer. So the natural question is, right, whether containers are still usable in restricted environments. And the answer is actually yes, but this is often through a different tool called Aptainer, which used to be known as Singularity. And some of you might recognize this if you're familiar with doing work on HPC systems. So it was created in response to exactly the kind of concerns we just discussed. Unlike Docker, Aptainer was built from the ground up for multi-user environments. Its design assumes shared infrastructure, strict security boundaries, and centralized system management. One of the most important differences is that Aptainer does not require elevated system privileges to run containers pulled from Docker Hub. So this single design choice eliminates many of the security concerns that make it difficult to get Docker approved by your IT department. So this is essentially an option for those of us who want to use containers, but we can't get Docker on our systems. So the way this works goes like this, right? Aptainer runs containers as the user who launches them rather than as a privileged service. From an IT perspective, this means containers follow the same permission model as any other user processes, and that makes them much easier to audit, control, and monitor. Because of these design choices, Aptainer is commonly approved on HPC systems and environments where Docker is restricted or prohibited. And it's really an intentional solution designed to bring container benefits to these environments with stricter security protocols. So Aptainer lets labs keep the advantages of containers, reproducibility and portability, while also aligning with the existing security policies. And nevertheless, it's important to note that Aptainer is really for running containers, not building them. So you may be relegated to what's available on Docker Hub or Quay.io, etc. You're not going to be able to really build your own containers with Aptainer. So up to this point, I've mostly talked about running existing containers. This slide kind of shifts the focus to why you might need to build your own container rather than just pulling something from Docker Hub. Public container registries like Docker Hub are incredibly useful, but they don't always align with your organizational requirements, and sometimes they don't have what you need, right? So many of these pre-built images are based on older operating systems. In regulated environments, there are often requirements around supported base operating systems, which, means that, well, we might have to build containers ourselves if we want to use them. So by building your own container, you can start from a base operating system that is explicitly supported by your organization. That gives you control over security updates, lifecycle policies, etc. And I'm not going to go into building your own containers in depth, but once you have a supported base OS, you can even use MiniForge as your base image to give you a flexible and reproducible way to manage software, right? So this approach combines the isolation of containers with the package management strengths of Conda. Another common issue with pre-built containers is version availability. The exact version of a tool you may need might not exist on Docker Hub, or it may be bundled with other software you don't want. So in some cases, older versions that are critical for reproducibility are no longer maintained in the public images. So MiniForge actually allows you to pin exact software versions inside the container. So that means you can explicitly define not just which tools you're using, but which versions, right? And that makes these updates intentional in the future if you want to update things. So building containers with MiniForge lets you align bioinformatics software with both scientific and organizational requirements. So, letting everybody know this is a real option for you too. All right, so just a quick recap before we move on. I don't have that many more slides left, but at the simplest level, we have system installs, things like apt-get, pip, or building from source. They're easy to get started with, but they're limited in terms of isolation and reproducibility. Conda gives us more flexibility, right? It allows us to isolate tools per project and manage dependencies more cleanly, which is why it's so widely used. And containers kind of take this even a step further. They focus on reproducibility and portability by packaging software together with its runtime environment, making it much easier to move workflows between systems. So the key theme really throughout this whole section, right, is that security policies at your public health institution really shape what's possible for you. In many cases, the best solution isn't one tool, but a combination, Conda inside containers, containers run with Aptainer, or different tools at different stages of workflow. So now that we understand different ways to manage open source software, the next question is how we actually use all of this stuff efficiently, right? But before we move on to the next slides, maybe we can just pause here for a second, see if anyone has any questions about software management, containers, or security constraints. And if not, I guess we can move forward. But feel free to put something in the chat if you have a question or comment. All right, so it doesn't sound like anybody has a question or a comment. So I'll just keep going then, right? So up until now, we've been talking about software, how to install it, isolate it, and make it reproducible, right? But software alone doesn't actually give us reproducible analyses. Even if everyone is using the same exact tool and version, that doesn't guarantee the analysis itself is reproducible, right? It also depends on how these tools are used and connected. So most bioinformatics analyses are not a single command. We develop pipelines, right? They involve multiple steps, quality control, pre-processing, reporting, etc. So those steps need to run in the correct order. If something runs too early or too late or with the wrong inputs, the whole thing can fail entirely, right? So as soon as you scale your analysis, more samples, more data, more compute, we have all these new kinds of failures that might appear. Jobs fail halfway through, files go missing, resources run out, things like that, right? So in public health settings, these issues matter because analysis often need to be rerun, audited, or shared across labs. Not to mention, right, a potential outbreak surge. So in that scenario, you might have many, many samples to analyze that you have not, and you're not going to be able to do that on your Windows workstation with WSL, right? So this is really where this whole idea of workflow orchestration comes in. So believe it or not, you can really think of workflow orchestration like an orchestra, right? So in this metaphor, each bioinformatics tool is like an individual musician. Each one is very good at a specific task, aligning reads, calling variants, etc., etc. But on its own, it doesn't produce the final result. The analysis itself is the performance, right? So to get a coherent result, every tool has to run at the right time, in the right order, with the right inputs, right? And workflow engines really act as the conductor in this scenario. They do the work, they don't do the work themselves, but they coordinate when and how each step runs. Where they differ is syntax, ecosystem, and how they execute jobs. Some are more script-like, some are more declarative, and some are designed to run across many different types of infrastructure. In practice, the choice of workflow engine is often driven by community adoption and infrastructure. If your collaborators, pipelines, or platforms already use a particular system, that often becomes the deciding factor, right? So the one that we use here that I'm going to talk about in the next slide is Nextflow. And I think this is what most people who use this type of orchestration for bioinformatics use. There are other options out there, but this is what I know best, so this is what I can speak to. So workflow engines like Nextflow are not about managing individual tools, they're about managing the process. We've already talked about software in depth. Something like Nextflow focuses on how tools are connected, how data flows between steps, and how the overall analysis is executed. Once those relationships are defined, Nextflow takes responsibility for running steps in the correct order. If one step depends on the output of another, Nextflow ensures that dependency is satisfied before moving on. So this becomes really powerful as workflows grow more complex, right? It can run, Nextflow can run independent steps in parallel. It can retry failed tasks, and it can resume workflows without starting over from scratch. And those are all things, especially when we're scaling up, that are really important, or if there's something like an outbreak surge, right? So Nextflow can launch steps using Conda environments or containers, depending on what's allowed by your infrastructure and security policies. For public health workflows, this means analyses are easier to share, rerun, and audit. The logic of the analysis is captured explicitly rather than just being hidden inside these scripts or manual steps that we might be putting together to do these types of workflows ourselves. And this kind of takes us to serverless workflows, which are kind of the future of where all of this is going, or maybe we're already there, right? So the other thing you can do with Nextflow and some of these other orchestration engines, right, is use serverless workflows in the cloud. So the term serverless can be kind of misleading, right? It doesn't mean that there are no servers involved. There are definitely things running on compute infrastructures, but what it means is that you are no longer responsible for provisioning, managing, or maintaining those servers. Workflow engines like Nextflow can take advantage of serverless execution models. In this setup, Nextflow defines the workflow logic and the cloud platform handles the compute infrastructure, right? So instead of running on a fixed cluster, compute resources are created only when they are needed. So as your workload increases, the system just scales automatically. So the examples shown here, AWS Batch, Google Batch, and Azure Batch, these are all services that provide this kind of on-demand managed execution. So I've been calling it Nextflow, but so everybody's aware, Nextflow has rebranded as Sakara. So we use Sakara Tower to execute our serverless workflows here. And you can submit tasks to these services without you having to directly manage the underlying infrastructure. That being said, they still operate with organizational constraints, cloud access, data governance, security approvals, etc. All of those controls still apply, but these workflows are, when you run the containers, they're ephemeral, right? They're there for a minute and then they're gone. So this is how our setup is configured currently for Nextflow Tower or Sakara Tower here at Wadsworth. And it works something like this, where we have the Wadsworth Bioinformatics core. We write code in Nextflow, which utilizes container. We push that code to state ITS GitLab, where that code lives. And we also configure Nextflow Tower with state ITS GitLab and Google Cloud. So when we have an end user in a lab who needs to run a workflow, they can go to the easy user interface, and then let's say they're running a workflow for, I don't know, something like TB. So they go into the lab, they go to the user interface, they run the workflow. Tower will grab the code from GitLab. It will then say, assuming the first step is genome assembly, based on the number of genomes or FASTQ files in the initial folder, it will scale up. So if you have, whether you have 12 genome assemblies to do or 25, it's going to spin up 25 different virtual machines, one for each, and do all of those at once. So you're not waiting to do them one after another. And then it's going to put them all into the same folder. Cost as having your virtual machine on for 2 weeks while you're trying to assemble all of these genomes. On top of that, once the files are moved into the cloud storage, those virtual machines are shut down automatically and disappear. So you're no longer paying for them. So it's a very cost-effective option, and it lets you scale really, really, really huge when you have to. Okay. All right. So, how do all these pieces fit together, right? At the foundation, software management controls what runs. Tools like system installs, Conda, and MiniForge define which software and which versions are used. Containers sit on top of that and control where the software runs, right? They define the execution environment, making it portable and consistent across systems. And the workflow engine operates at the highest level, controls how everything runs, right? The order of steps, parallelization, retries, and scaling. And surrounding all of this are our institutional policies, the main thing that probably drives the way we do things. Security requirements, IT controls, and compliance rules shape what's possible at every layer here, right? So there's no single right way to do these things. The best solution is the one that fits your data, your workflows, and your institutional constraints. And the goal isn't to adopt the newest tools, it's to build workflows that are essentially reproducible, secure, and sustainable. So that kind of concludes my talk. And what I hope you take away is that these tools are not competitors. There are different ways to use them based on your constraints. They're really complementary layers that solve different problems. So when you align software management, containers, workflows, and policies, reproducibility kind of stops being a challenge and really becomes a design choice when you understand these choices. So that kind of concludes my talk. Thank you, everybody, for taking the time to listen. And with that, I'm happy to take any questions or hear how these challenges look in your own environments. Feel free to share your own experiences or not, right? So thank you very much. So I have a question here from Tony, which is, do you have a Sakara Enterprise license paid for by Wadsworth? So we do. So we use Tower with Sakara, and we do pay for licenses. We have about 12 licenses, I think. Those licenses were a lot more expensive. They've kind of come down in price, but that is something we do pay for. With Tower, I will say that you can still use Nextflow on a virtual machine in the cloud to spawn Google Batch jobs serverless. You would just be stuck in a command line environment to do that. So you don't need a license to do that. But if you're going to scale up where you have this nice user interface that your lab members can use to run their own pipelines, for that, you do need a license. Amy says, really informative talk. I appreciate that. Thank you very much. Anyone else? All right. Well, thank you, everybody. I really appreciate your time. Oh, Curtis has a question, right? Is the Wadsworth Center using StaPH-B Docker images? If not, is there anything the StaPH-B could do to enable you to use them? Curious if there's... Yeah, so we do use them for some things and not for others. The big issue we have right now is the operating system issue in containers. Our IT policy and our IT people are telling us we can only use containers where the underlying OS is fully supported. So we've spent a lot of time over the past year building our own containers with where the underlying OS is Ubuntu 24 to give us the longest lifespan. So I think if you're going, if the StaPH-B group is actively building containers, I would say try to get those underlying operating systems up to date as possible. So I think it's kind of put us in a position where we can only use about half of the StaPH-B containers, which are the ones that have the up-to-date operating systems. All right, thank you, Curtis. Well, I guess that's it. Thank you, everybody, for your time.
Yeah, thank you so much. That was really interesting.
All right.
Have a good day, everybody.